From Microbe TV, this is TWiP, This Week in Parasitism, episode 262, recorded on July 11, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about parasites. Joining me today here at the incubator in New York City, Daniel Griffin.
Hello, everyone.
Welcome to the incubator. Welcome back. You were here a few weeks ago.
Yeah.
Right. Yeah. But the occasion is that we have a guest. But before that, let's bring in from Glasgow, Scotland, Christina Nowla.
Hello, everybody. I'm a bit envious for not being in the incubator. But it's nice here too, so.
Our guest today is from right here in New York City, Chuck Knirsch. Welcome.
Thank you.
What's your affiliation? Do you have one?
So I'm doing quite a bit of board work now on the Task Force for Global Health in Atlanta, which is going through some hard times with the USAID cutbacks. Also, something that might be interesting to you, which is I chair the Wolbitos board, which is the World Mosquito Program board in Brazil, where we grow up mosquitoes, Aedes aegypti, in Wolbachia, and hope that it stops transmission of dengue, Zika, yellow fever.
So you're going to be releasing those in Brazil?
It's already going on. The releases are going on. We actually have impact data. So maybe in the future
nice
virology meeting, we can talk about it.
So what are your thoughts on the trashing of U.S. public health and science?
It's hard. And I guess the way I'm trying to deal with it is to understand what I can do to do more. And part of that is to understand people that think that public health is not important, that international health is not important, to understand that and try to engage it in a civil way. I think, when you feel like yelling and screaming and having a seizure and things like that, and at times I do feel that way, I don't think it's going to be productive to get into a dialogue with people to try to build consensus. And that's what I'm trying to do in some of the board work I'm doing. But it's very, very hard because the impact is catastrophic right now in low-income countries. And it's only been a couple months.
Right, we've had cuts to Gavi. We've had cuts to USAID.
PEPFAR is not functioning. Vincent, there are hundreds of millions of dollars of donated drugs and vaccines sitting in ports that are not being moved to where they need to go. That needs to be fixed. And so I think, with some constructive discussions and pressure, forming other coalitions, finding new ways to do something without this major funding source that comes from USAID is what I think about. And of course, what we're going to talk about today, a neglected tropical disease, will be impacted by these types of cutbacks. Then,
of course, here domestically, we're witnessing the destruction of NIH, CDC, FDA. We have an anti-vaccine guy heading HHS. This is also problematic.
I used to have, and I will continue to try to have faith in the Senate. This is where, understanding, with seasoned politicians, to put people in positions with agendas that you could have predicted from reading the Project 2025 work, and then without any sort of way to negotiate, to understand their position and negotiate to a position that maybe isn't what it was, but there's something else. I'm not saying it's in the middle, but some other consensus. I like transformative change. I don't think one or two people that don't have experience in health are the people to unilaterally make the changes without, So anyway, I mean, it's going to be a long haul. I'm playing for the long game, and I believe in public health. I believe in pro-health measures. I also believe in change, and I'm willing to engage in those types of discussions.
Okay. I wanted to get your thoughts because we talk about it all the time, right?
Yeah. I actually, my wife's sister married a diplomat. He wasn't a diplomat when they got married, but he is now, and he's actually coming tonight on his way to Mogadishu. And so we'll be talking a bit, but been a lot of things going on with the State Department today. I think over 1,000 layoffs were announced. So they were told, everyone come into work today because we might want to fire you. are talking about moving USAID under the State Department, which might be okay, actually. But yeah, because it needs to function. I mean, USAID does a tremendous amount throughout the world. And yeah, you can't have millions of drugs just sitting there.
It's also, so the Task Force for Global Health has a lot of ex-CDC people, a lot of ex-Carter Center people, some people have worked in other parts of the U.S. government, and then some Africans and people from some of the low-income countries. And these are early warning systems in these countries for the emergence of new diseases, often viral. And, I think everybody's clear that we would have had many more cases ago. I mean, a Columbia University emergency room physician who had been doing, volunteer work in Liberia got Ebola, was fine when he arrived here. We would have had many, many more of those if we didn't have the early warning systems in places like Liberia or Ebola. So I'm not trying to be an alarmist, but we're building the capabilities and the human talent in those countries to do the work themselves. And frankly, many of the things that have been cut by USAID, we had a plan to sort of phase into country ownership of programs by 2030. Okay, we got 5 minutes after the inauguration to do it. Not, the 5 years that we really think we could have done those transitions and have these countries, have their own public health organizations stand up on their own and do the work and collaborate with us and protect each other from emerging diseases. Anyway, we're a little off topic. I'm sorry to go down that pathway.
No. I think
it's a passion.
Public health.
And it scares the heck out of me what is going on right now. Frankly.
Yeah, no, I think our listeners care deeply about this. And I think that was probably, for me, as an important point is there was a plan to transition over, and it was a 5-year plan. And, and there's so many reasons to do this, right? Some people argue it was diplomacy, it was soft power. But there's also a moral, right? I mean, we have a lot of resources. We have the ability to make a difference in the world. So there's a moral aspect too. And it just seems like this sudden, we're stopping today without any warning, without allowing things to get in place. There's a certain cruelty, I think, we're seeing there. We don't care. And that's not the message we want to send to the world about the United States. We're not a people that don't care. We're a people that care.
We care deeply.
Yeah. And I just think, unfortunately, there are a few bad actors that are really thwarting what I think, most people would like to see a better world. Most people would like to see a better world because they care, because they're decent human beings. But also we want a better world because there are no borders. I mean, you get from anywhere in the world, 24 hours later, you're here. And you're here with Ebola. You're here with whatever. We saw that with COVID. Very quickly, diseases spread. And if we don't have that expertise, those warning systems, there's even a selfish aspect to making the world a better place because we all live in it.
Yes. And I thank you to you and Vincent for some of the work you've been doing with the lay press. I think it's really important for you to get out there. And again, in any ways that we're good at, because you both are very, accomplished communicators through, TWiP and other venues, TWiP today. And I think it's really important for you to be, and I sent you a note last week after you were talking to the New York Times, Daniel. So really nice work on your behalf.
Yeah, thank you.
Yeah.
We do have a paper today. It's paper time. And Christina is going to help us discuss this.
I think it's Daniel's paper.
I know. I wasn't going to say anything. I'd like to put you on the spot there. See a little color come into your face.
She got nervous there. Yeah.
I just feel like flushing. I did read it.
No, I have to say this. I hope everyone's ready. This is going to be a delightful, exciting paper, and it's a theme. I have to say because we're going to do a little bit of a tribute to Dickson. But this was a way to get Dickson sort of hot under the collar, was to talk about how parasites influenced mammalian behavior, right? And toxoplasmosis was always when I get very upset when I would bring that up. So he wanted to talk about the hydrenosome and hydrogenosome and all these other things. But
well, they're cool things.
They are very cool, yes. And if you're stuck in a Prius with Dickson for many hours, you'll hear about them. But let's get into.
He would eventually come around, though, Daniel. Maybe it was your persuasiveness, but you would, and then he would go off on a tangent. And
I like to poke the bear. That's part of the dynamic. But all right, so the paper we have today is in PLOS, Neglected Tropical Diseases, entitled "Does Schistosome Infection Affect Behavior Through the Gut brain axis. And it ends with a question mark, and I think we're still going to have a question mark at the end, but I think we have some exciting data here. So I'm going to have to put on my glasses here to read the authors and go through a little bit. So,
hey, before you do that, what's on your
what's
bow
on
tie?
my bow tie today? So it is a Friday.
Sexually transmitted
It's a
disease.
Sexually transmitted disease. It is a purple spirochete. It's not purple in reality, but what's spirochete?
Syphilis. Huh?
Syphilis, yes.
The great masquerader?
Yeah, the great masquerader. Okay, so we have a number of authors here. Leah Combrink, Johanny M. Spahn, Alex Perrett, Thomas Mehara, Brittany Hyun, Dana Parker, Jennifer Johns, Michael Bluen, Kathy Magnuson, Michelle Steinauer, and I apologize for all the mispronunciations there, but they're from around. We've got some folks here from Arizona, Tucson, Arizona. Got some folks here from Lebanon, Oregon, France. More folks from Oregon, Corvallis, Oregon, and the Linus Pauling Institute in Corvallis, Oregon. So we won't be talking about vitamin C. I know people like hear Linus Pauling and want to do that. So let's start off with a little bit of background. So schistosome infection. Actually, Chuck, you want to give us a little background? What is this schistosome infection?
It's just a fascinating worm. And I sort of think of it in terms of life cycle, where the adult and female worm pairs live in the venous system and magically escape the immune system. And how they do that, I think, is still an area of research. The female obviously lays eggs, several thousand a day. And depending on whether the schistosome is in the venous plexus of the bladder, which is schistosome hematobium, one species, or in the plexus leading into the liver, schistosome mansoni, and some others like japonicum, the pathology is the eggs actually causing inflammation in those two organs, respectively. Eggs are also released through the GI tract, and there's an intermediate host of a snail where the further development goes, finally leading to cercarial stages, which reinfect humans through skin contact with freshwater sources.
So that's why you have to towel yourself vigorously.
This is that
right?
that episode, towel off vigorously. So the story is I'm with the same relative, right? At this point, my relative in the State Department is stationed in Malawi. And he's got 3 boys, married to my wife's sister. And then there's, my 3 children, my wife. So the 10 of us end up going to Lake Malawi. And I'm telling the kids, fresh water, sub-Saharan Africa. I'm sorry, guy. There's no swimming. We get there. There's a lot of local kids swimming in the water there. So again, we're not going, but it's just so hot. So we end up swimming. And then my advice to the children is when you get out, you must towel off vigorously because the cercaria penetrate as you get out of the water and it starts to dry. Now, I don't know how 100% effective that is. But
you weren't involved in a randomized controlled trial on that?
Did you have your drugs with you for treatment just in case?
Well, so interesting enough. So, my relative and his family, they were there for a couple of years in Malawi. So when they came back, they went through testing. You get treated with praziquantel just as So.
And the disease causes. Sorry, back to you.
Sorry, I was just going to, it's a good time of year to get duck schistosomes in Loch Lomond in Scotland, although they cannot penetrate, but you get a proper yucky little swimmer's itch. So those schistosomes are everywhere. Don't cause disease so much. Not here, but yeah.
So here, what they're going to be doing is they're going to be asking whether schistosomes, and they're going to be using a mouse model here, whether infection with schistosome can impact behavior. And they're also going to try to go into how might it do that? So they're going to have some ideas. So I don't know who does the editing, but those of you who have the paper in front of you, and maybe whoever's doing our YouTube can put up figure 1, because I'm just going to just sort of lay out what are they going to do here. And so it's sort of interesting. So they order up the mice, right? And you can order these from various places. I'm that nerd who, when you're up in Maine, like, I want to visit Jackson Labs, and the family's like, no, we're going to keep driving. Yeah, I was very excited.
It looks like a cool place.
It's not far from Bar Harbor,
read
right?
this paper. I wanted to go there.
Yeah. So you order your mice, and then they show up. And now what they want to do is they want to sort of make sure the mice all have sort of a similar microbiome. So they try to, as they say, homogenize the microbiome. We'll just cut to the chase. It doesn't work so well. They sort of get this different. So you got to sort of try to,
quite some effort to do that, though.
Yeah, they do, but they fail. That doesn't.
Yeah, they do.
Or I should say, they don't succeed. How about that? It's not that they fail. They don't succeed, but there's still success here. And then they go ahead, and there's this nice, you're sort of following this timeline of the design. Then they're going to expose them to the schistosomes. Then they're going to do these fecal collections. There's going to be a praziquantel, that antiparasitic I mentioned, treatment. They're going to do a whole bunch of testing, and I'm going to go through what are all these different tests: the open field, the novel object, the marble burying, the fecal collection, the Morris water maze. And they're really going to be asking a couple things, comparing infected to uninfected mice. Are there behavioral differences? So I'm gonna actually talk about what are these different ways that they're going to look at mice behavior. And I think for some of our This Week in Neuroscience, our twin listeners, they're going to be familiar with some of these mice things. I thought it would be fun. So talk a little bit about what they are, because then when we look at the data, people will say, oh, I get that. This makes sense. So the first behavioral test they're going to do is the open field test. And the idea here is you've got these mice in these plastic box arenas. And if they're theoretically, here's the idea. If they're really anxious, they're going to kind of just sort of sit there in the center, not really going to be wandering much about. But if they're less anxious, they'll be more exploring. Instead of staying in the center, they'll be all around. So think of that as heading out into the open areas, not as much time spent in the center zone. So open field is going to theoretically be a way of saying, getting infected with parasites, is that going to affect your anxiety level? Okay.
Daniel, I thought it was the other way around, but maybe I got my got my brain is boiling essentially. But I thought it was when they were in the outer boundaries that they were having higher anxiety levels.
So they say more time spent in the outer boundary indicative of anxiety or fear. So yes.
Yeah, okay. Yeah. Because I think that's the other way of what you said.
Yeah, no, it is. And it's really, it doesn't make sense to me, right? Wouldn't you think that if you were anxious,
you would huddle in the middle?
Yeah. Maybe if you're anxious, you're bouncing all over. I don't know. It's sort of, so that, correct. So we'll see that. So the metrics are going to be 3 metrics. And one of those metrics is the more time spent in the outer boundary, indicative of increased anxiety and fear. Total distance traveled, right, is supposedly indicative of healthy exploratory behavior, right? Comfortable moving all around. And then the permanence time spent in the outer boundary. The other, and we'll go through open field because we're actually going to look at the data on choose. The other is the novel object task, and this is going to be looking at memory-based recognition of novel objects. So they're going to give them sort of a time to get used to these objects, and then they're going to wash them all off and make sure they're clean, and they're going to see how they react to the novel objects being introduced into the scene. And the whole idea here is the expectation that if mice remembered the familiar object, they would spend more time checking out what's new, right? So we're going to have any impact there. Marble burying task. That almost makes sense. It's like the, obsessive, they have this compulsivity or anxiety disorder. So the more time they spend like burying stuff is supposed to be a measure of this anxiousness. And then one of my favorites, the Morris water maze task. Mm-hmm. So Vincent, you're familiar with this, right?
Yeah,
they talk about
they
this all
use
the time.
this in Twin all the time. Yeah.
It's kind of cruel, actually. Right. I don't know if our listeners know, but mice do not like being in the water.
Right.
They do not like it. I mean, I don't know if it's, but this is like this torture for the mice, but spatial learning and memory. Because what you do is you've got this area with water, and then there's a clear platform. And the mice learn where that platform is.
It's submerged, so they can't see it.
They can't see it. They got to remember where it is because it's just a little bit under, but they get on there and get their bellies up out of the water. So they really want to get there. Now, it's not horribly cruel because if 60 seconds go by and the mouse hasn't found it yet, they're going to lift them and put them on there. So it really is just...
They do learn. When you put them in on a second test, they will find it much quicker than the first time. Right?
Exactly. And here we're going to ask, does having a parasitic infection affect that? Okay, so we've got our... And then we're going to talk about fecal microbiome. But let's go to the next figure, right? So we've got our time scale laid out. And then the next figure, I mean, I'll mention it, but I just, it sort of seemed like what they were showing here is the more you expose, the more cercaria you expose the mice to, the more worm burden they end up getting. Right. So basically, worm burden was higher in mice that were exposed to 75 cercaria compared to like 50 cercaria, for instance.
I think they also looked at the pairing, don't they? They looked. I think that was also proportional to the number of pairs. So if they had more in cercaria, they would also have more, not single worms, paired up worms. I don't know how relevant that is.
I wasn't sure. I mean, it made sense to me too. I mean, the more worms, the more worms you have, the more are going to sort of match up male and female. Whenever they teach the schistosome course, they always sort of get all excited about, this wonderful thing where you've got the male and the female and one's wrapped around the other.
In copula.
Yeah, it's in copula. Right. So I'm not sure this is so much this warm, cuddly, taking care of each other as they're like just constantly having sex. Right.
Yeah.
Right. So I sort of think of this more as like a pornographic thing than like this wonderful romance novel vision. But anyway, okay, that's me.
They're careful to evade the immune system, whether they're covering themselves with albumin or whatever mechanism they're doing.
That's true. That's true.
I always wonder what happens to the non-paired worms. Do they just die, or do they just hang about and maybe hopefully find somebody? I often think about that, sadly.
So they do try to find each other. And do you know how a male and a female schistosome find each other?
They're just delightful.
Yes.
That was Dickson's.
Yes, they find each other delightful. Okay. So now we move to 3, and I think 3 makes sense. They basically ask, infected versus uninfected. Even though we talk about how much immune evasion is required for these worms to survive, these parasites to survive, is there some activation of the immune system? It does look like there is some cytokine response to being infected with the parasites. And IL-23 tends to be like a huge difference, right? Some difference in IL-17, right? I mean, same sort of level of statistical significance. IL-10 is up quite a bit. So figure 3 is really comparing cytokine concentrations. The black bars, if you see those, are the infected, the gray bars, the uninfected. So we are seeing some sort of cytokine response. And now we're going to do behavior. We're going to see.
Just not with TNF-alpha was what they noted, right?
Mm-hmm.
Yeah, and there was no association between the worm burden and any of the cytokines, so it didn't really matter how many schistosomes you had. I think that made no difference in the cytokine profile.
Kind of interesting, right? Like we would sort of think of the more worms, the more so.
But they didn't really have that many. I think they had on average about 3 pairs. So, they were quite similar because they were the ones that they infected with 100 cercaria, they treated with praziquantel, so they would have a reduced worm burden, I guess, to make them less sick. So I think they all had around 3.
Yes, that might be an issue. Yeah. Maybe they didn't have the range like that heavy infection that we might see. Okay, so now the open field.
Daniel,
test.
Would you expect these elevated cytokines to last the lifetime of the mouse?
No. I mean, I would think once you treated them, once you got rid of the schistosomes, you would expect the
right. But if you did not treat them,
and you had chronic infection, no, I would
see it elevated. Yeah.
I mean, they might.
Well, we know that the eosinophilia is our marker clinically to pick it up is elevated. So I would imagine that that's probably due to some interplay with chronic stimulation and chronic inflammatory state.
Okay.
And we always talk about part of it is exposure to the worm, but part of it also exposure to the eggs.
Yes.
Mm-hmm.
So,
yes, that's critical.
And this is actually a fairly short infection. So, right. I mean, if we had carried this out longer, we might see, so that's interesting too, right? This is a very short time because we might get different results. What happens if over time, now you're starting to see the egg exposure? What does the egg exposure do?
Mm-hmm.
All right. So now what happens here? So now we're going to go to figure 4, and we're going to look at the open field test. And the mice infected with schistosomes spent more time in the outer boundary than uninfected mice. So what does that tell us? Remember, because I had this backwards.
So it essentially means that they're more anxious.
Mm-hmm.
Interesting. So it is kind of interesting, right? So the infected mice are more anxious. So and it's a pretty significant difference, statistically so, infected, uninfected. So spending like almost 85% of the time in the outer boundary versus about 80% uninfected.
Mm-hmm.
And then the other is sort of, entries into the center. How often are they heading back into that center zone versus scurrying all about anxious? So it does, this does seem to be a difference. It looks like, at least in this particular approach, it looks like being infected with schistosomes is making mice more anxious. This is sort of like when Vincent expected you to present the paper, right?
I was immediately scurrying into the outer boundaries of my little room.
Yeah, I saw you there starting to move away from the screen. But
I was thinking, oh no, I missed the memo.
Now, this next one is kind of interesting, right? And they're gonna revisit this in the discussion. So what they're going to tell us is that they're going to do this novel object task where they put this novel object in, and the mice infected with schistosomes had a higher memory retention after 1 hour than uninfected mice, but no difference at 24 hours.
Does this mean they're more curious? I couldn't quite wrap my head around that, actually.
Well, I think their interpretation, right, of this mouse test is the idea is you put something novel in there. And if they recognize like what was there before and recognize that, hey, there's something new, they're going to spend more time checking it out. But if you don't know, the two things could be novel because you don't even remember what you saw before, then you're going to just spend equal time just checking them both out. At least that's their idea.
Mm-hmm.
So, because mice are curious. And so the whole idea is, hey, there's something new. What's here? Let's go check it out. But if you don't remember what was there before, then everything's new. And you won't see that sort of discouraged. But it is interesting. 1 hour, 24 hours. Because 1 hour you see the difference, but 24 hours it's gone.
So they had greater memory retention after an hour than 24 hours.
Yeah.
So this is a beneficial thing, I presume, right?
Sort of interesting, right? So schistosomes make you anxious, but then you remember things, which could almost be bad. Like if you're some traumatic thing, you're going to remember it and you're going to be more anxious. And so... or is there some advantage to the memory? Maybe I get schistosome infected right before that big exam, but only an hour before.
Well, memory is useful for averting dangerous situations because if you've experienced a dangerous situation and then later on you remember it, you might be able to avert it. But this is what we call the evolution of gist memory. Mm-hmm. When you first have an incident, you have a very precise remembering of what happened, but then within 30 days, it fades and becomes just the gist. It's a very vague outline of what happened to you. And so you could meet someone, and then 30 days later, you meet someone else who looks like them, and you'll say, didn't I meet you? Because your memory has faded. Mm-hmm. And so the reason it does that is to protect you, because if it's too specific, you won't respond to a slightly different situation, right? So maybe that's what's happening here. It's just
like I came in and you didn't have a turtleneck on. I could see your neck, and I wasn't sure who that was, right? But then that memory generalization kicked in. I'm like, I think it's Vincent. So at this point, I mean, before we go on,
it's too hot for turtlenecks.
It's too hot for turtlenecks. So at this point, I'm sort of thinking, and you sort of, as you go through, it's like, does this make any sense? Like, right, because we're going to go on to a couple more. But is there any evolutionary advantage to schistosomes? Selecting for schistosomes that might elicit this behavior.
So the only improved memory is 1 hour.
Yeah.
And then it goes away. So I'm not sure what...
You're not sure, and maybe they're a little more anxious. They're a little less likely to get, because once the schistosomes get into the mammal, you want that mammal around, right? You want that mammal to survive. You want them to keep, getting more eggs back out into the environment. So if whatever little sort of variation in you keeps your host alive a little bit longer, they're a little less anxious, they're a little more anxious, or a little, less likely to get eaten by that crocodile or that hippo.
Do mice get schistosomiasis? It's just a model for it. In the wild, do they?
I think it's just really a model. I don't know. Do you want to Google
I'm
this?
I'm going to Google that, yeah.
Right.
So the thing is that if humans have the same responses, which we don't know, but let's assume they do. So what is the purpose of us getting anxious when we have a schistosome? And it doesn't seem to have any evolutionary purpose.
Yeah.
Right.
I guess the only thing I was thinking is if, because you're getting this by the water's edge, right? Mm-hmm. Crocodiles, there's hippos, there's predators. So, instead of you being the first of the kids that goes running and plunging in, you're going to sort of wait a little bit back, and that's has an advantage for the schistosomes because, you don't get eaten by it's the first kid in that gets eaten by the crocodile, right? So, when we're at Lake Malawi, there was a lifeguard there. And I asked him, I said, so you're sort of standing here on the beach. Can you swim? I mean, you're the lifeguard. He's like, oh, no, I can't swim. I was like, well, then what's the point? What are you doing? He's like, I'm watching for crocodiles. And when the crocodiles get too close, I yell. And then I tell everyone to get out.
Gosh.
So, so I was like hanging out on the beach with this guy while my kids were out there because, maybe I was schistosome infected from prior trips and they're just reckless out there at risk of getting eaten by crocodiles. So, all right, we'll circle back to speculation. So now they do the next test, and this is the marble bury test. And basically what they're going to do is they're going to be saying, what are the difference in as far as the number of marbles buried, either fully buried or partially buried. And that's going to be, forget which figure that's in, but I remember them not really seeing any difference there.
Yeah, no difference.
Yeah, there's no figure either. I guess that makes sense if it shows nothing.
Nothing to see here. So, I mean, I probably still would have stuck a figure in, but nothing to see here figure. Now, the last is the Mars water maze task, right? So this is, you've got this pool of water. There's that hidden clear platform that they remember where it is, but they can't see it. So they've got to remember. And you're gonna ask a couple questions here. One is like, how much are they going to wander around before they get there? How much time is it going to take them to get there? And this is going to be in figure 6. So let's go to figure 6. And it's good that they did it several times because when you compare the infected to uninfected, so the corrected cumulative distance, right? So think about it, it's like how much do they have to travel around before they find that platform? And it does look like there's something of a difference in the infected, right? Their corrected cumulative distance, it's a little bit more. In some.
So they have to swim. If they're infected, they have to swim more. They don't find it as easily.
That seems to be the thing. So they're sort of claiming that maybe there's some impact upon this spatio-temporal memory. So a little harder for them to remember where the platform is if they got infected. Which is a little bit in contrast, right, to that novel.
To the other one, yeah.
Yeah, the other one where they're like, no, they maybe this sort of enhanced memory for.
This is not significant, so maybe we should just say that, right?
Yeah, because it is. If you look through, you're not.
The error bars overlap.
They all overlap, yeah.
So they tortured the poor mice for nothing.
Hey, NIH wants to eliminate animal experiments.
Oh.
So they're going to use this as ammunition.
Exactly. But I mean, I don't know. I think if you watch the mice, they're unhappy, but they're not actually.
Yeah, they're fine. Yeah,
they're fine. Okay. And now they get into the area, which really, is more, I'm going to say, more of a correlate, because I'm having a little trouble buying the causal connection here, because I feel like there could have been some more experiments. But now what they're going to do is they're going to look at the microbiome differences. So they're going to look at the schistosome infection causing this shift in the gut microbiome composition. So you're going to see a bunch of figures here, and they'll kind of run through that. So we've got basic,
And the reason why they're doing is that because they speculate that it's the changed microbiome that might actually promote the changed behavior that we've seen.
Right.
So what did you think? Were you intrigued? Did you find it compelling? Was it a little rise to the level of interesting?
I don't know. There's not really anything conclusive, really. Unless I completely missed the point. I mean, I think it needs a lot more work. But let's hear your discussion first, because maybe I was too hot when I was reading the paper.
No. I mean, I sort of was on the same level. Like they talk about, changing the abundance of different organisms. But I think for any of this really to be brought home for me, I would have had to see them do like a fecal transplant. Yeah. Establish this new, and then see that, hey, if we just change the fecal, microbiome, we saw that increase in anxiousness. We saw this, 1 hour change in, in novel memory. And I think we're sort of left hanging. We're left, by the way, there was a change in the gut microbiome, which we're not surprised, but lots of studies before have shown that parasites, change the gut microbiome. But what does that actually do? Is that just an epiphenomenon, or is that driving the behavior change?
They would like to say there's a connection between the behavioral changes in the microbiome, but you can't really do that, right?
Yeah.
I got the sense from the paper that they have built this model. They've studied it pretty carefully. But I couldn't tell, where this would go in the future. Because presumably you would take this and you want to translate it to some clinically useful, application in other species, including us.
Yeah.
So, would we really do an intervention with a microbiome to prevent some of the neurological? Let's say that's a given that these tests in mice do translate to humans. They are used, some of these tests, as screens for pharmaceuticals. They reference a few papers. Would you then, if you then believe the microbiome piece, would you do that instead of do better work with praziquantel to eliminate it? I guess the naysayer would say, yeah, but you're going to get resistance to praziquantel someday, so we may be left to, altering the microbiome. Mm-hmm. I don't know. I think if I were a grant reviewer, and it may be my ignorance in knowing what this team will do next.
Yeah.
So I'd love to know that before I say I'm a grant reviewer looking at maybe some chemistry to develop a better praziquantel drug or something like that versus investing in a microbiome alteration.
I mean, and maybe this is how you get your next grant, because I'm left hanging. And if they said, okay, now our next experiment is going to be, we're going to change the microbiome and show that alone is what drives the change in behavior. And then you start saying, it's always that translation, like, how does this help people? Maybe people that are anxious have a certain, microbiome, and if we change that, then they're going to be less anxious, less compulsive behavior.
Transplant.
Yeah, maybe we start.
Study you already suggested. They may have already done it, or they're planning to do it. So that would be interesting.
Yeah, and that would buy you your next grant, because that's going to matter, right? We've got lots of anxious, compulsive people with all these things going on. If changing the microbiome can lead to better outcomes for these folks, then the problem I have, though, and this is going to be, a great paper. I mean, just authors, if you're listening, that's why we're talking about it. So don't, because I think sometimes when when someone's critiquing your paper, you feel like, hey.
Right. No, it's a very nicely written paper.
Yeah,
very logically follows through.
Yeah, very well done. Excellent work. One of my issues is that the mouse gut is so different than the human gut, right?
Yeah.
So, but it's tough because, how do you then study this in humans? So, and then even interventions, right? It's hard to say we're going to change the gut microbiome in people. We're not going to infect them with schistosomes. You almost have to sort of wait for an opportunity to see similar.
I think when we're looking at such broad impacts in mice, I mean, even when you look at narrow things like immunology. We know that mice don't really translate all that well to, say, non-human primates. And we know that the monkeys always lie when you get into the human studies.
Oh, yeah.
And so, but you have to start somewhere. So I think good for them. Nice work. And it certainly made us think of some future experiments. And given they're the domain experts, I bet you they have 10 more to do in the future than we've suggested already.
Yeah.
So their conclusion is we did not find a direct interaction between behavior and microbiome composition in schistosome-infected mice. So there's nothing there. And they said maybe they're not related, or maybe it's too correlated to tease it apart with this model. So you need to do something else.
Yeah. I suppose, it's scientifically really interesting. And, I think there's a lot of value to experiments or studies like that, because it can teach us a lot, but maybe from a public health perspective, we have other really good measures to maybe educate people to, be cautious around fresh water, like toweling off, or we can improve water and sanitation. So we do have really good measures, but I think actually from a scientific curiosity perspective, I thought that was a really interesting and actually also quite fun paper.
So they in the introduction they make the point at saying, many people are infected with schistosomes, and in kids, studies have shown that the infection impacts the behavior of them, deficits in school attendance, school performance, and memory, and increased behaviors, hyperactivity, aggression, conduct problems, anxiety, and withdrawal. But I don't know if they have other infections.
They're likely to have soil-transmitted helminth infections, I would assume, and, maybe other ones too.
But you could do a study in humans. You could take, I don't know if this is ethical, but you could take a group and treat them and not treat another group. But probably you can't do that, right?
They do these. The UK folks do these rollouts, right? Like we're gonna roll out a deworming program here. But you just can't do everything at once. So you see like here, and then you roll at the next place. So you really have kind of a natural control.
It's a legitimate design to have a control group that has delayed treatment in effect that's not planned for.
Then you could look at these deficits and see if they change with treatment, right?
But I suppose the effects will be maybe a result of chronic long-term infection. So I don't know, maybe you wouldn't really see much of an effect.
Well, I wonder in the young kids now, we're sort of doing a reverse rollout, right, with the cuts to USAID. If we are no longer supporting these programs and you start to see a rise in infections, a lack of deworming, are you going to start to see more anxiety, more aggression, more violence? Like, are we going to end up having, so many other untoward consequences that we didn't really think about?
That's a study that needs to be done, right?
I mean, it's kind of like occurring like before our eyes. It'd be a question of like collecting that data by pulling back with the USAID, by pulling back all these treatments. Are we going to see more conflicts in these areas? Are they going to spill over? Do we get global instability? Because it only takes one angry person to do something they shouldn't do before things start to escalate.
I don't know. If you have some kind of infection, then you get anxious, and that can affect your behavior. But if these kids probably don't know it, right?
No,
I don't think they.
And so that's interesting. But I would say that many other things could affect their behavior as well. And I don't know if these studies were able to. It's an observational study, so I don't know that they could do anything about that.
And it's mixed in with, malnutrition. And the thinking up until I think, and then acknowledges, I think, in the discussion, is that there's, permeability issues with the gut when there's chronic inflammation, other parasites, and that there's leakage of nitrogenous and other substances across that may be causing some of this effect. But again, kudos to this group for setting up a model. Be interesting to see where they go next.
Yep.
Yeah. We're looking forward to your next paper.
Thank you, Christina.
You're very welcome
for that presentation.
You actually jumped right in and did a lot there, Christina. So.
I did. I always read the papers in some detail, but I didn't write specific notes, which I would do if I present myself. I would have a pile of papers to look through. But yeah.
Okay, what's up next, Chuck?
I really thought we should talk a bit about Dickson. More in kind of a celebration. And I thought I would start by just talking about when I first met him and how our relationship grew over time. And please interrupt at any time, because Vincent, you knew him for a really long time, and Daniel, the amount of work that you did with Dickson in the last decade. So I moved to New York from Montreal to do my residency, and Dickson sort of oversaw the parasitology parts of the clinical laboratory, and he worked with Dr. Elner, Harold New, and others. So there was always sort of once a month a malaria case, and Dickson would come, throw on his white coat and show up on the wards and be part of the team. And as you know, he has this gregarious, larger than life personality, and he was noticeable. I was interested actually in doing international health, and so Glenda Garvey had said to me, "Well, go talk to Dickson. He's approachable. You don't have to make a appointment; just show up in his lab." And so I did that. And I was thinking going to London school, and he says, "You got to do that." And wrote me a letter of recommendation for a grant I tried to get, but I think the stock market crashed. So I ended up moonlighting a bit to pay my way to save some money to get over to London. But Dickson, started off, did an undergraduate degree at Fairleigh Dickinson, and then started working as a lab technician, and then moved into a master's program at Columbia. And his mentor was Harold Brown, who we'll maybe speak about a little bit later. Then went to Notre Dame, and then to Rockefeller University, where his mentor was Miklos Mueller, where he was working on trichinella. And so Dickson also, by the way, since he was a kid, had a fascination with spirals. And so, Vincent had been very kind to Dickson, I think it was maybe during the pandemic or earlier, and set up, Dickson loved photography, and set up, I think, both a high-resolution printer, but also a screen, which Dickson has worked into many of the books that he's published over time. But his time at Rockefeller was notable. He said, one of the things that was the highlight was going to the cafeteria and sitting down with, Nobel laureates, just, really brilliant people. And one of the people, and this will explain part of why, he was so into environmental, René Dubos was somebody that he would sit down with when he was at Rockefeller and talk the environment, talk science, talk about whatever. And so, I think Dickson's second, third, and fourth career has been something completely different than studying Trichinella spiralis, which, for a long period of time, he had a number of R01 grants. The other thing, as a clinician that I was impressed. So after coming back from London school, I started my ID fellowship. Dickson was like, "Okay, now you're going to come teach the course with me." And I said, "What do you mean?" So Dickson for 30 years would do a 1-hour lecture, and we'd have a 2-hour lab. Actually, all of Harold Brown's specimens that he kept, so nematodes, we would roll out as macro and then do the histology with the students with that. And so I think it was just a remarkable course, which I didn't have in medical school. I don't know, Daniel, whether in medical school you had a formal lab component to the parasitology lectures.
No, we didn't. And actually, down at NYU, where I went to medical school, a number of the parasitology teachers were visiting, and actually Dickson was one of my parasitology professors at NYU.
So you can imagine that 3 hours times at least 8, lectures. There was some faculty interested in getting some of those hours, especially for the lab part, and just turning it more into a lecture course. And Dickson was constantly sort of grousing about that. And I think also, as a educator, if you get a Teacher of the Year award, that's sort of one of the crowning joys, and right up there with any other sort of citation award you would get. If you look at Dickson's CV on Parasites Without Border, there was a number. I think he may have won that award 6 times or something, several years in a row.
It's true.
It's at least 6, maybe more.
Yeah.
And Linda Lewis, who was the dean of students for quite a long time, a very gifted clinical neurologist, actually nominated Dickson for the National Teacher of the Year award, which he won.
Wow.
So I think it just shows you the breadth of what he was doing in the laboratory. So I want to focus on a paper because Dickson, when he was still on his R01 grants, when I was a resident, and I think was on the cover of the 4th edition. I mean, when I walked into the incubator, Vincent took me over to Dickson's, sort of the books that Vincent's keeping in care of and trust of Dickson. I think the 4th edition had the Knirsch cell.
That's the blue cover, and it's got the...
No, that's the 3rd edition.
That's
when
third
Springer
edition.
was still... So the first edition that when Dickson's, desktop published, actually that's the edition I joined, the 4th edition.
Mm-hmm.
We'll have to check it after this podcast to make sure I'm not making the story up right now. But I'm pretty sure it's the Knirsch cell. And this is the way Trichinella actually works with skeletal muscle to embed itself. And Dickson's work with Virginia Capua and then this Ramona Polare, how does it do this? How does it create this vascular arcade to survive in? And actually isolated vascular VEGF, it doesn't roll off the tongue, vascular endothelial growth factor, which at the time was being studied in some labs in Boston, for potential oncology uses. So if you can inhibit vascular, you could starve the tumors, their blood supply.
Which we do clinically to this day.
Which I worked on that drug, actually. Sutent is for kidney cancer. So I just, that's just a little tidbit on his science. So that was what he was excited about. So there is a certain place I used to have, and both you used to have lunch with Dickson, which was Coogan's. There's actually a 1-hour documentary, and highly recommend that documentary because it talks about this Irish pub in Washington Heights with a large Dominican immigrant population and sort of the center of activities, including half marathons and community organizing. I think Juan Lynn Miranda, actually, his family would regularly celebrate birthdays.
They did a fundraiser when Coogan was having trouble during the pandemic. Yeah. It kept it open for a little while, but yeah, there's a connection there.
So I would have lunch there with Dickson at least once a week. And, we'd talk about whatever. It would be sports, science, how things were going in our families, how things were, interesting patients, the course coming up, after a course lecture. And, if we were there in the evening for dinner, we'd walk out and, at this time, the garbage bags would be sort of piling up on the streets and, for the morning pickup of garbage. And Dickson started saying, this is unacceptable. I mean, why can't we sorry, some sort of an insect just flew in. I hope it wasn't Aedes aegypti with all
Well, it's the incubator where we've got a colony going.
You've got a mosquito going on. Excuse me, let me just swallow it. And it's tasty, actually. So, Dickson, we start musing about this, saying we need, and this is when the locavore, momentum is coming. And Dickson went down to the lower campus. So for folks that don't know, the Columbia system, the health campus is up on 168th Street. The undergraduate campus is down more in the 110s in that area. And Dickson actually put out a proposal to engage architecture students on the concept of skyscraper farming. And, so I brought along, actually, I was looking for a couple books. I brought along The Vertical Farm, and actually this one where he thanks us, you and I, for the several edits. And so I want to read, though, that so a Nobel laureate that won the Nobel Prize for his work on ivermectin, Bill Campbell, I think explains Dickson better. And this is a foreword to a book. So in the VEGF story, Dickson was, and it relates to our first topic, which is schistosomiasis, was fascinated by how the male and female worm could live in the vascular system for 6, 7, 8 years, laying eggs the whole time, and and evade the immune system. Why can't we learn from this and wanted to affect transplant medicine? And so he wrote a book on these. It's basically called People, Parasites, and Plowshares, Learning from Parasites. And the foreword was by the Nobel laureate. I don't think he'd won the Nobel Prize. But
Not yet. No,
not yet.
Before he won it.
And so I'm just going to read this because these words, the whole thing's worth reading. I'm just going to read a little bit of the front. And I have to read it because to paraphrase it wouldn't do it justice. And so, first of all, there's a quote from Ben Jonson that says, "Oh, your parasite is a most precious thing, ... from 1607, Ben Jonson. And so now Bill Campbell writes, "No less a genius than Alfred North Whitehead was of the opinion that one is unlikely to have new ideas after the age of 60. But one can still make good use of the ideas one already has. It is no secret that Dr. Dickson Depommier's genius has been more than 60 years in the making, yet in his conversation, new ideas burst forth like bullets from a machine gun. Many of his friends have experienced the phenomenon firsthand, an experience that his former colleague, Dr. George Stewart, has referred to as being Dicksonized. I think we miss being Dicksonized.
Yeah.
The phone call that would be, the Knicks last night, that after 45 minutes was like the continuum of the life experience. Let me just stop there for the game. Maybe Daniel, Vincent, you knew him.
No, I always made a point if I was going to talk to Dickson, if it was going to be on the phone, I made sure I had like a long car drive ahead of me and I had blocked out, because there was never a quick call to Dickson. It always was. And it would just start going and this explosion of, what about this? And so very dramatic and novel ideas. And
I met Dickson in 1982 when I went to Columbia, and he was, I don't remember if he still had his lab at the time, but he taught a lot. And he was, as you said, he was a very good teacher. And I would see his lectures and they were really good. And he was also a wonderful guy and easy to talk to. So then in 6 years, it was many years later, 2008, I wanted to start a podcast. And I thought of him because I knew he was very enthusiastic about teaching. He was very eloquent and could explain things and had broad knowledge. So he and I started TWiP. In 2008, it did well, and then we started TWiP after that, this podcast. And then all the others followed. But Dickson was on TWiP and TWiP. He also started a urban agriculture podcast for a while where he interviewed people about this indoor vertical farming idea. And he was on TWiP until just a few months before he died. So he was an essential part of my career. And I don't know who else I would have got to start it. Now, when Daniel showed up in Steve Goff's lab, Dickson at that point had moved into our outer office. And Daniel used to come by and he'd talk to you, right? Because he's a chatty guy.
Yeah. So I'm on my way to see Steve, and and here's this individual there, and we start to talk, and then it occurred to me, I know this guy, and and we made the connection that in 1990. He had taught my parasitology, taught one of the parasitology lectures at NYU. And I said to him, not only do I remember you, but I can even tell you how you got to NYU and what you had for lunch. And here it was 2013. It was 23 years later, and he had made that much of an impression. Yeah. And he had taken the subway down and stopped en route to have sushi. Which was very appropriate for a parasitologist, right?
So he decided to bring Daniel into TWiP. And he said, this fellow Daniel, he could really help us on TWiP, because we had gone through all the parasites and we were starting to do papers, but he thought we needed something else. He said he could do a clinical thing. And so I credit Dickson with bringing Daniel onto this program, which I think has revitalized it and made it much more popular. So I just said, okay, I trusted him that it would be fine, right? I trusted him that he thought you would be fine, that the idea would be fine, because I'd had so much interaction with him over the years. So he was truly an interesting fellow on TWiP. He would always go off tangent, but really interesting. People loved him. He had interesting picks of the week. He would go through jazz and music and photographs, and really contributed a lot. I think a lot of people miss him. But they're all there. Those podcasts, you can go listen to them. You can listen to the very first TWiP, the very first TWiP, Urban Agriculture, only 30 podcasts there. So it's there for your listening pleasure.
Yeah. And on Parasites Without Borders, we have all those videos, right, that you filmed.
So we did two sets. I did one with Dickson, and then after you arrived, we redid them, right?
And we added this little clinical vignette.
Yes.
So each chapter has a video, and then
there are
vignette.
There are two sets of videos on YouTube that you can find if you go to the Microbe TV channel. And that's all what we did with Dickson and Daniel. Yeah, it's great. It's good stuff. Dickson had my philosophy. He wanted to teach people for free, right? That's what we want to do. That's what Parasites Without Borders wants to do. That's what Microbe TV wants to do. That's why it made sense for us to join in this incubator here. We want to give everyone an education without having to pay for it. we're giving it away. Because what we know, we have many years of knowledge. We want to give it to you because that's the way it works. Just like the U.S. used to give money to pay for drugs, and we don't do it anymore, but we will continue to do it.
Yeah, what is our motto at Parasites? Getting knowledge to people in places that need it the most. And I think right now I know a country that really needs knowledge, real knowledge, not just facts that someone invents to support an agenda.
Yeah, that would be the United States, right?
Is that where we're going?
Well, I mean,
Yes.
this is terrible. I think many people in the U.S. are recognizing that this is a problem, but it's hard to pull people out of power, right? The way the structure is right now. But slowly things are happening. You saw all the medical societies are suing.
Yeah.
And now in the Senate, there's some movement to prevent Trump's cuts to NIH because they don't think it's right. And the Senate, of course, is who gives the NIH money, right?
Right.
So maybe
we're not far from Moynihan Station, and Daniel Moynihan's. I don't know if you know his famous line: "Everyone's entitled to their own opinion, but they're not entitled to their own facts."
Yeah, right. That's absolutely right.
You can't make up the facts.
True. Yeah, no, it's absolutely.
All right. Are we going to have a pick hero, Chuck? You think?
I think it would be fitting, since we've been thinking positively about Dickson, to talk about one of his mentors. I'm sure Dickson had many mentors and different degrees of mentorship, but he talked a lot about Harold Brown.
Always.
And so Harold Brown was a professor. He'd gone to Duke Medical School, and then he came to Columbia in 1943. And had several initiatives that he started. He started a school of public health at the University of Puerto Rico. He actually started in 1951 a medical elective in the tropics. If you think about it, that's so far ahead of his time now that you can do these things as well. And Parasites Without Borders has actually supported some different type of travel type of electives. And then this was written, actually, one of the last things, I was looking back on emails from Dickson over the last couple of years, and this is one of the last things he wrote. He'd written this email with this attachment to both Daniel and I, saying he wanted to have Harold Brown, held up, in our book, which we've just finished, and hopefully we'll publish shortly.
Yeah, we're about to print some copies, so I just need to do a last few little edits, and then we'll be doing our first printing of the 8th edition.
Is it on the website yet?
It's on the website now. I need to make a few
changes,
pagination corrections. Apparently, I did not catch when the output came that the page numbers are a little bit up. We're going to fix that this weekend. Tomorrow, I feel under the gun.
This weekend.
It's my first day off in 20 days of work. So my first day off, meaning my opportunity to focus
with
on
no air conditioning.
Yeah, when the air conditioning is going to work. So I will have air conditioning and time. That's all I need.
All right. It's TWiP 262. You can find the show notes at microbe.tv/TWiP. You can send questions or comments, TWiP@microbe.tv. And if you enjoy these podcasts, we'd love to have your support. We don't do advertising. We depend on listeners' donations to keep these going, to keep the lights on. Go to microbe.tv/contribute. Chuck Knirsch, our guest today on various boards and organizations. Thank you, Chuck.
It's a pleasure being with you both, and with Christina from Glasgow.
Daniel Griffin is at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Thank you. And everyone, be safe.
Christina Naula is at Glasgow University. Thanks, Christina.
Well, thank you. That was fun, as it always is. A highlight of my Friday evenings.
I'm not going to say anything.
I'm being honest.
I know. I'm just kidding. Well, we're all here on a Friday night, too. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiP, and Ronald Jenkins for the music. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back in 2 weeks.
Another TWiP is parasitic.